In this video, we're going to look at eukaryotic DNA and we're going to look at prokaryotic DNA and we're going to compare the two. We're then going to have a look at how aspects of DNA in mitochondria and chloroplasts can be evidence for the evolution of eukaryotes. So, you've probably heard of the word chromosome before. There's a definition there for you to write down. Now, a chromosome is just the genetic material that has been packaged up into a structure, and the structure is called the chromosome. So, on this slide you can see representations of all of the chromosomes that are present in humans. Now, you may be familiar with chromosomes or refer to the structure of chromosomes as that X shape. Now, that X shape means that the DNA has been replicated. So, in actual fact, a chromosome is more accurately represented as a sausage-like structure like this. And humans have pairs of each chromosome. So we get one chromosome from mum and one chromosome from dad. So in our human genome, we have 23 pairs of chromosomes, including our sex chromosomes down here. Now, when you see the whole genome represented like this for any organism, we call that the karyotype. So this is the human karyotype. And an important note for you there is that organisms can have a large variety of sizes and genes on chromosomes. So a chromosome could have from 10,000 up to 1 billion base pairs on a single chromosome. Right, so this is how DNA is actually packaged up. I want to draw your attention first over here to this organelle. Now, I should know by now that this organelle is called the nucleus, and within the nucleus we've got our genetic material. You can kind of see that squiggly line there. And then if we zoom in, we have our genetic material all coiled up and packaged into a chromosome. We can see that this chromosome has that X shape, so the DNA here has actually been replicated, ready for cell division. And if we zoom in a little bit more, we can see how that singular strand of DNA has been coiled up into this rope-like structure. And then if we zoom in even further, we can see how here is actually that tiny strand of DNA, and it's wound around these purple balls here. Now, we'll talk about them in a minute. And then if we zoom in even further, our familiar structure of the double helix. As we go in even further, we have our base pairs here with our hydrogen bonding happening there. C pairing with G and T pairing with A, as is always the case. Now, on this side over here, we can see the same sort of thing, but in a bit more of a structured way. Again, our double helix, and here are those now yellow balls that the DNA, the strand of DNA, winds around. So these balls are called histones. They are proteins, and their purpose is to package DNA. Now, when you get DNA wound around histones like this, we call them nucleosomes. And here you can see nucleosomes all packaged in together. So this part here represents just a small section of one of these coiled up strands here. And then that coils up even further here. And we can see that this part over here represents just one small section of a chromosome. And now these are some true images from an electron microscope, or from a microscope, and there's been some dyes put in here so that we can see very clearly some different structures, and we can see our cytoskeleton here, and then this is our nucleus. We know this is the nucleus because it's stained, and this part that's stained in the middle, this is the genetic material, the DNA in there. We can't really see any distinctive structures in there, and that's because it's unwound, right? The cell in this phase has the DNA that is not packaged up at the moment, and when the DNA is in that form, we call it chromatin. As it starts to get packaged up, we can see our sausage-like structures beginning to form over here, and then even more distinctly over here in this phase. Right, now here's another transmission electron micrograph for you to have a look at. Again, microtubules here part of our cytoskeleton holding the chromosomes in place. And then here are our sausage-like chromosomes over here. So these chromosomes are packaged up. The DNA is all packaged up. And in eukaryotic cells, that's what they look like. We have multiple linear chromosomes in such a way. This slide here is a little bit busy, but just another representation here of how the DNA is packaged up in these nucleosomes around histones. A bit of a closer look at the nucleus here. We can see our nuclear envelope with our nuclear pores and then electron micrographs of those, just so you know what they actually look like. And something interesting to ponder on over here, have a think about, if the chromosomes contain the genetic material and they reside in the nucleus, then how does the rest of the cell get access to the information that they carry? I want you to ponder on that for a little bit. Now, this is a bit of a look back at some of our previous learning. I want to draw your attention to the part that's been highlighted in yellow. Now, eukaryotic cells contain a nucleus and they have DNA within that nucleus, which are bound to proteins called histones, and they have multiple of these chromosomes that are linear. So they are the aspects of eukaryotic DNA that are important to remember. And here it is written down for you so that you can take some notes on it. Now, prokaryotic cells have a single chromosome and it's circular. And over here, it doesn't really look like a circle, but it's all wound up, tangled in and around together. This is actually one singular circular piece of DNA, and it's sort of localized in a region within the bacteria called the nucleoid region, but it is not a nucleus, important to remember. And again, another representation just to be able to compare what it actually looks like under a microscope. Over here we can see our nucleoid region, not always easy to spot. And then over here what it looks like if we're going to show it diagrammatically is our nucleoid region. Now, another important aspect to remember about prokaryotes is that yes, they have one single chromosome which is a copy of the entire genome, but they also have other little circular bits of genetic material that don't contain the whole genome, and these are called plasmids. So they are small bits of genetic material that are also circular, but they do not contain the entire genome. They are called plasmids. And here is a summary table for you. Good idea to get this one down into your book. Now, here is an interesting aspect of mitochondria in particular, but also the chloroplasts. So mitochondria and chloroplasts have their own small bit of circular DNA. You might have heard of mitochondrial DNA before. Well, it's actually curious to note that they represent, or they resemble, bacterial DNA very closely. So the theory goes that mitochondria and chloroplasts at one stage in evolutionary history were their own prokaryotes, and then at some stage they were engulfed by another prokaryote, but then the relationship was mutually beneficial because the mitochondria gave the other organism energy and the chloroplast gave the other organism carbohydrates from photosynthesis. And so then they replicated and carried on to form early eukaryotes that had the first organelles, mitochondria and chloroplasts. We're going to revisit this idea a little bit later on, so don't worry if it's all been a little bit quick for you, but just interesting to note. And here that is in a little bit more detail if you would like to have a bit of a look or a bit of a head start on the idea of endosymbiosis.